to study. Basically, we take just one astrocyte and we study whatever the RNA, anything, whatever you need. But the one cell we mostly use for RNA studies because it's not much protein there. Pony, this can you see this slide?
Yes.
Yeah. So, okay. See, this is the yeah. This is very interesting. See, this is from the rat brain. Yeah. Yeah. This cell. Just no. This picture it is to show this is a cell with this scale. The same scale in the two. Can you see that this one astrocyte and this one astrocyte, but the scale is same. That means the distance, whatever the cell taken from the tissue is the same, but this is so small and this is so big. The very interesting part here is this is from rat brain, this is from human brain. See how much difference between these. So they are all not.
Excuse me.
Yeah. So there's a great difference between rat brain and the human brain. The astrocytes are not the same size between the animals. They vary between whatever the brain they have. So in here I'm going to this is from the rat brain and this is from the human brain. So difference. Yeah. So there is something going interesting. Why they are different, we don't know. How that will be, considered for any study.
Excuse me, Jacob. Again, the slide goes off.
Can you say this slide now?
No, your face only. Your face only. Hello. Okay, I'll tell. Okay.
Okay.
Just a minute. Yeah. Sorry for the interruption.
Yes, no problem. I will go to the next slide. So what?
Excuse me, your slide is not visible. Our outsourcing department asked to send your slides to them so that they can show the slides. Tbml.com. Can you send or you can produce the slides now? Someone give the comment. Sir, pin your presence, sir.
Madam, excuse me, madam. This is Gobinathan. The same name, sir presentation name, Shyamala Devi Nagajanu. அந்த நேம் பின்னாடி அப்படியே ஸ்க்ரோல் பண்ணீங்கன்னா இருக்கும் லாஸ்ட்ல. அத பின் பண்ணீங்கன்னா சாரோட ஸ்கிரீன் விசிபிள்ல இருக்கும்.
எப்படிமா then what I have to do? Okay, Shyamala Devi.
அந்த நேம் அப்படியே பின்னாடி லாஸ்ட்ல இருக்கும். அத ஸ்க்ரோல் பண்ணுங்க.
Shyamala Devi.
எஸ் சீக்வென்ஸ்ல பாருங்க, எஸ் சீக்வென்ஸ்ல. அத பின் பண்ணீங்கன்னா சாரோட ஸ்கிரீன் தெரியும்.
பண்ண சொல்றேன். அவரோட பிக்சர் வந்து ஸ்கிரீன் போட Jayakumar, start. We'll see.
Yeah. You guys stop talking, please, for a few minutes and let me know what the problem is.
Okay.
So I'm still sharing. It says I don't have a problem in sharing. So
in the beginning, we can able to see your pictures, the slides. But after that, only we can able to see your face. The third slide you can show that the astrocyte. After that it was not visible.
Yeah.
Jivanandam here. After pinning Shamala Devi, etc., now I could see view the slides. Thank you. After making the pin, now I could see I could view the slides now.
Yeah,
it's clear now.
Suggestion given by Gobi, I think.
Yes, it's clear now.
Is it clear to you, sir?
Yeah, after making the pin. Now we could view the slides. Now it's clear. It's clear only by pinning it.
Yes, Jaykumar, you can continue now.
Okay.
Okay, continue. I think everyone able to see now.
Yes.
Yes, thank you, sir. Thank you, sir. Dr. Jaykumar, you can present now.
Okay, so I will go to the next slide, and so yeah, no problem. I'll be able to manage. So here, as I mentioned earlier, there are these astrocytes always come. Yeah. So I hear a lot of sound, so I don't know what.
Hello, Jaykumar. Hello. Hello. Hello, Jaykumar.
Yes.
Yes. You continue, please.
Okay, thank you so much. Yeah. Okay. So yeah. Yes. I will continue. So hello? Yes. Can I continue?
Yes.
Yes, please continue, sir.
Whatever I spoke is coming back. Yeah. So can I go? Hello, Dr. Puni. Can I present now? Yes. Can I present now?
Yes, present.
Okay. Thank you so much. Maybe if you all please keep quiet for a few minutes unless you have any problem or something, so that I can move forward. Okay. So this slide is very, very important. So this has some basic informations. encephalopathy. Encephalopathy means any brain problem due to dysfunction of other organs, maybe from liver or maybe kidney or maybe anything, lung or heart or yes. Everyone hear my voice?
Yes, sir.
Yes, we hear.
Okay, wonderful. So I will continue. So
Yes, please.
yeah. So what happened? I'm talking about encephalopathy. Why I'm talking about encephalopathy? So astrocyte, particularly the Alzheimer type astrocyte is particularly present in any conditions that have encephalopathy. Encephalopathy means the brain dysfunction due to other body organs like liver, lung, or heart, or kidney, or whatever there. So what happened when there is a encephalopathy? So what happened? With the encephalopathy? There are two conditions in general happening in the human brain. Yes. Okay. So what happened in the okay. Okay. Pony, can you hear now? Hello? Hello? Okay. I didn't present until such a time because whatever I presented before, they are all eating. So that's what happened. Okay. So what happened? So, Pony, can you hear my presentation or no?
Yes, we couldn't hear Jay, but the person there asked me go for our presentation mode and bring it to full screen. Then you
Okay.
present.
Okay. I will do that. I will do that 1 second. Can you see now?
Yes.
Can you see now?
Yes, yeah. Yes,
Okay.
please.
Yes. Something mess up. The program
we
is
can
mess
hear.
up, but what I can do, I
Oh.
continue and then stop wherever I can.
Okay, it's very interesting. You go ahead.
Okay.
Can you start from this encephalopathy, Jay, Dr. Jay?
Yes.
Okay,
Yes.
please. One
Yes.
second.
Yes. I will continue. So what happened?
Yes.
The encephalopathy, they occur in two. Yeah. It's a lot of background noise, so I'm not able to. That's why.
Participants, kindly mute your mic, please. Participants, kindly mute your mic. And now everything is clear. You can able to hear and you can able to see the video also, the slideshows also. Participants, kindly mute your mic, please. Yes, Jacob,
Okay.
everything is clear
Wonderful.
now? Okay,
I think it's better than before. Okay, thank you so much. Thank you so much. So I'm going to continue now.
Yes, from encephalopathy. Excuse me, Dr. Jay, again, the sound is not clear. Jay?
Okay. So, Ponni, what happened? Whatever I spoke before, they are all repeating again.
Okay. Is it okay, Jay? Jay Kumar? Jay Kumar, sorry for the interruption.
Ponni.
Yes, yes, Jay.
Something screw up, Ponni. Because whatever I spoke before, they are all coming now. I'm not talking.
Oh, okay. Now, don't worry about them.
Yeah.
Start.
Did you hear me, Ponni?
Yes, I am here.
No. What happened? I just want to make sure.
Yes, Jay Kumar, I'm able to hear you.
Hello?
Yes. Any problem, Jay Kumar?
Excuse me, Jaykumar sir.
Yes. What happened? Whatever I spoke before, they are all repeating.
Yes, sir. And background, you opened your YouTube also, sir. Close that one.
This one and go to YouTube.
YouTube. See, sir, in your window.
Okay, I will try. Okay.
Just YouTube only. Closer to YouTube. Hello, sir. Ma'am, Ponni madam.
Yes.
சார் அவரோட கம்ப்யூட்டர்லயே யூடியூபே ஓபன் பண்ணி வச்சிருக்காரு. ஒரு 40 செகண்ட்ஸ் டிலேல போயிட்டு இருக்கு. அதனாலதான் அவர் டிஸ்டர்ப் ஆயிட்டு இருக்காரு.
அவரை நான் க்ளோஸ் பண்ண சொல்லணுமா?
நான் சொல்லிட்டேன், க்ளோஸ் பண்ணிட்டு வரேன்னாரு. யூடியூப் ஓபன் பண்ண வேண்டாம்னு சொல்லுங்க சார்.
நம்ம இன்னும் ஒரு 45 minutes இருக்கு. Jayakumar, you stop the YouTube and come to Google Meet. Yeah, we are running short of time. Yeah, please stop. Stop it. Okay. Yeah, that the thing is, the YouTube link is started after 45 minutes. That is why that is going on. You stop this. You stop the YouTube and come to Google Meet. Then you continue. Immediately. But most of the participants, they are going out. Participants, please. Yeah, we can see now. We can able to see you. You stop the you. Yes, okay. Okay, right. Yeah.
Sir, unmute your mic, sir.
And Jayakumar, now we are on Google Meet.
Jayakumar, sir, please unmute your mute button.
Bala, you give instruction to sir.
Jayakumar, sir.
Dr. Jayakumar.
Sorry. Dr. Jayakumar, sir, please unmute your mute button, sir.
Yeah, can you hear now?
Yeah, yes, sir. Thank you, sir.
Okay, can you see the screen now?
No, sir, we are not getting a screen, sir.
Yeah, I'm okay from my side. I have nothing problem. So
Bring your slide, Jayakumar. You start.
yeah, my slide is there. So I'm showing right now my slides.
Okay.
So it says that I'm presenting. So can you see that now? No. Now what happened? I'm in full screen right now.
Yes, you're in full screen now.
Can you see the screen or no?
Not the picture, only your face.
Excuse me, Ponni madam.
Yes.
Actually, I'm Lakshman. I have an idea.
Yes.
Sir has to send his PPT to anybody's mail ID, madam. On behalf of sir, somebody have to show the PPT. He has to talk only. In this way, we can solve the problem.
Okay.
Simply, he has to send his PPT to anybody. For example, your mail ID.
Okay. Right.
You have to show the PPT on behalf of him.
Okay.
He has to talk only. In this way, we can solve the problem.
Okay, okay. Then I'll, okay. Jayakumar, can you get the suggestion?
I'm 1 second. I'm sending it because it's a huge file. I will try from my work ID.
But it is difficult to you?
No. You may not be able to receive it. You may not be able to open. That's the problem.
Okay, that's the problem.
I'm sending it a new message. What's your ID, please?
My ID, christyponni@gmail.com. But the outsourcing person asked me to send.
Christy Ponni, 1 second. I think I have your email. 1 second.
Jayakumar, don't send it to me. Jayakumar, don't send it to me.
Okay. Give me the ID.
Tbml.com.
No. You have to spell out.
MA.
Okay.
DHEAS.
Okay.
Tbml.
Okay.
.com.
.com.
Yeah.
No, this is not email.
Yeah, just I'll ask Bala.
Ma'am, Gmail. Ma'am.
Gmail.
Gmail. Ma'am, MATHS. TBML @gmail.com.
Okay, Jay?
Yeah, I'm attaching files.
Okay.
Madam, that outsourcing person has to show the slide. Sir has to talk.
Okay,
That's
sir.
all.
Okay.
Yes.
Thank you.
Yes.
Thank you.
Jayakumar, did you send it?
1 second is uploading. Yeah.
Okay.
Yeah, I sent.
MSN, good morning.
Morning.
Good morning.
Very good morning.
Very good morning.
Okay, we continue.
There is a technical flaw. We are rectifying it.
Yeah.
Yes.
Thank you.
Oh, hi.
Jay, whether it is uploaded?
No, I sent her. I sent.
Okay.
It's a huge file. It's a huge file. My file might reach through. So he has to look for the cloud.
Okay.
I can tell how big that one is. Yeah, that's a 62 MB file. So he may be able to receive it.
Mail வரல இன்னும் 62 MB file சொன்னாங்க சார். 62 MB. OneDrive linkல அனுப்பிட்டாங்க.
That's a cloud connection through work university. So for me it's easy. I'm sorry, I don't know how much time you're going to take.
Yesterday also asked me, Jay. Yesterday also they asked me to send all PPT again.
No, this is a huge file. One of the problem, and I have a lot of restriction through UM,
Okay.
So a lot of files that's not published already. So not published, some published, so they might create any problem. So but it's okay right now. It's okay with me. So why you guys using? You guys not allowed to use Zoom there or what?
No.
Yeah, the Zoom is easy. This one, there are some problems. So you may not have any problem with the Zoom unless if you doubt that website or something. So I strongly recommend go for Zoom and ask the IT person who are taking care to monitor where they are copying and all those things. He can put a block,
Ma'am, one more option is ma'am, one more option is ask the host to display slide in his laptop and use for audio from his mobile. It will be more easy. Huge file, it's not easy to transfer to another mail, ma'am.
You give the suggestion to Dr. J. He understands Tamil. You can talk.
Yeah, no problem. Tamil. பேசுங்க.
சார் உங்களோட ஃபைல்ஸ் வந்து லேப்டாப்ல இருந்து ஸ்லைட் ஷேர் பண்ணுங்க. உங்க செல்போன்ல இருந்து ஆடியோ ஆஸ் a பார்ட்டிசிபெண்டா என்டர் ஆகி மியூட்லயே வச்சுக்கோங்க. அதுல இருந்து ஆடியோ மட்டும் நீங்க பேசுங்க சார். லேப்டாப்பை நீங்க பாத்துக்கலாம்.
Slow. I'm sorry, I can't follow you. Slow.
அது வந்து சார். ஓகே சார். Thank you சார்.
இல்ல மறுபடியும் சொல்லுங்க. நீங்க ரொம்ப ஃபாஸ்டா பேசுறீங்க. மறுபடியும் சொல்லுங்க.
உங்க லேப்டாப்ல வந்து கூகுள் மீட்ல ஷேர் பண்ணுங்க ஃபைல் மட்டும் சார். உங்களோட ஸ்மார்ட் போன்ல இருந்து ஆடியோ வந்து நீங்க பேசுங்க.
I got it. Okay. 1 second.
We will try this also, Jay.
லேப்டாப்ல ஸ்லைடு ஷேர் பண்ணுங்க. எல்லாருக்கும் தெரியும். திரும்ப மீட் போன்ல இருந்து.
Jaikumar.
Yes.
Jaikumar, now the slide is open here. The people is connecting.
Okay.
Will you able to present now? Okay. Bala, இது clear ஆயிடுச்சா?
வர சொல்லுங்க மேம். அப்ப ஆடியோ கேக்கும்.
மொபைலா?
ஆமா மேம். மொபைல்ல வந்துட்டு
Yes. I'm in the mobile audio. Both I have. Very nice.
Ma'am.
Okay. Can you all see the slide now?
Yes, it's clear now. Yes, it's clear.
Okay, I will go from now. Thank you. Oh my God.
You kindly rush it off, Jay, please.
So I'm trying to scroll down and I'm not able to scroll down. So I have to do something.
No, sir. You have to guide me to
sir down arrow key use பண்ணுங்க. Sir keyboardல இருந்து down arrow scroll
down arrow. Okay. I will try. No, keyboard down arrow doesn't work.
Otherwise enter key you can use sir. Enter key தட்டுங்க. Next slide போகும்.
Okay, அதுவா போகுது. கொஞ்சம் maybe slow probably. Wait பண்ணுங்க.
Okay sir.
Yeah, it is a little slow. I will manage it. 1 second.
Excuse me, sir.
Yes.
Sir.
It is very slow. So.
PowerPoint நான் play பண்ணிட்டு இருக்கேன். So you have to guide me. You have to guide me in which slide I have to stop and which slide I have to move.
Okay, so from this slide, go next one. I will tell next. Okay. Okay. Go ahead. Go to the next slide.
Yes, sir. Encephalopathy.
Yes, wonderful. Okay, I will go a little slow. When I say yes, next you proceed. Okay, So from here, you guys listen carefully. This is very important. I'm shifting to from the astrocyte to the disease. So you may be able to get some input from. So this slide basically what happened. Always disease can occur in two forms, one acute and another is a chronic. So in the encephalopathy, I said what is encephalopathy? Like brain dysfunction due to any organ failure. That's what the encephalopathy. So I'm taking here in the whole slide with the hepatic encephalopathy. That's a condition where the brain is not working properly due to liver problem. So what happened in the liver problem? The people who has a liver problem, the brain won't work. I can give good scenarios. For example, what is that? Alcohol. What happened when you take alcohol? So when you drink the alcohol, mostly it won't hit the brain and it won't go inside the brain. But what happened? It make initially they make a liver fibrosis, and then later stages it become cirrhotic. They call us a liver cirrhosis. So in that condition, what happened? So during the liver cirrhosis is a very chronic problem. Some people, they have the disease for many years, for 20 years, 30 years, or 40 years. So during that problem, what happened? They have a mild to moderate brain problem, like a neurobehavioral problem or motor coordination problem or sleep problem. They have many problems. So that condition we call as chronic. So what happened if it is acute? When the acute can happen? That's the very interesting part that everyone, it's not for the scientific part, everyone should know basically. For example, if someone take the alcohol for 20 years, what happened? If he has an additional insult, for example, if you have hepatitis virus, particularly hepatitis virus, what happened that screw up the liver very fast, in a day or two. So what happened? What happened that time? The liver, jaundice is simply jaundice. When you take alcohol, if you have jaundice, within 2 days, without any treatment. So that condition called acute cases. Most probably this occur when there are so many complications like liver problem or a kidney problem together, or they call us multi-organ function. So that condition called acute HE, acute hepatic encephalopathy. So what happened in the acute hepatic encephalopathy? The cells which I am talking about, the astrocyte, they swell. So this is particularly interesting for every one of you. So what happened when there is, for example, if your liver is not working properly due to alcohol, or you taking some medication, for example, acetaminophen, you take a lot of Tylenol, right? I don't know what, paracetamol, for example. What happened when you take the paracetamol, the liver start to not to work properly because that screw up the liver. So what the problem when the liver is screw up? So very, very interesting that when your body make a lot of gases in the stomach, because the bacteria, all the end product of the bacteria in the digestion is so many gases. One particular gas, that is ammonia, that come to liver. Once it come to liver, what happened? It converted into urea, and then it go to the urine. That's what the way you get the urine, right? So if there is a liver problem, what happened? That ammonia will not be converted. So the next part where it can go, it bypass straight to the brain. So that's the important aspect in this particular disease. Once it go to brain, what happened? I showed you before. There are so many cells in the brain. Very interesting. The ammonia go only to the astrocyte. That's what the problem is. So why it is going only to astrocyte? That's evolutionarily developed. What happened? This cell has an enzyme glutamine synthetase. That glutamine synthetase, what happened? Convert the ammonia to glutamine with the help of glutamine. So once the ammonia go inside the astrocyte, that binds with the glutamate and become glutamine. When there's glutamine, you know that there's osmolyte. that bring water inside quickly. So this is the major problem. When water come, what happened? The cells start to swell slowly and become at one point the big swelling. So when you have swelling, what happened? That increase the intracranial pressure, and then you will go to coma and you go to death. That's what happened. In particular, very interesting. I told you, if someone take alcohol and if they have jaundice, this condition happen within 24 hours. So here, not only here, everywhere the practice is, they take to hospital and then they leave them to die. They don't treat anything, even mannitol, anything they don't treat. Everyone has to die. So how they die? The pressure, intracranial pressure increase because the astrocytes, they swell, they become increased brain edema, and then what happened? Start to try to pump more blood into the whole body, including the brain. That blood, what happened? That the blood start to burst everywhere. That's why if you see too much alcohol, if they have jaundice, the blood start to come from air, mouth, everywhere. That's the place they start to die. Yeah, I don't see the slide. I'm sorry. Yeah, the next slide, please. Can you make the next slide? So, can you enlarge it? Okay. So this is the slide I was telling. So I cannot, I cannot see in the big screen, but I can tell from here. So I know what happened. This is the slide I was telling when the ammonia bypass. They come to the liver and the liver won't be able to metabolize the ammonia. So then they bypass directly to the brain where it converted into glutamine. That's the glutamine bring water inside the brain and then it start to swell. It expand actually. So when it expands, they automatically die because there won't be any blood flow into the brain. So the blood vessels from everywhere start to break, and then you get blood from nose, ear, mouth, everywhere, and then they die. Even if you go to hospital, they don't treat anything. They leave you to die for a couple of days. This is what, there is no treatment at all. Next slide, please. Yeah, so this is one of the interesting slide which we have. The top one is the normal human brain. So here you can see these all grooves. They have expanded, and they have the grooves clearly. But when you go to the brain with acute liver failure, someone has acute liver failure. Here you can see that there is no such a thing because it expanded. So it has become bigger and bigger. It expanded. There is no any gap here you can see. This is the classical picture which we took from human brain. So next slide, please. Yeah. So see, this is also the same human brain here. This called HNE hematoxylin and eosin staining. Here you can see this. This gap, all this gap, they are all water inside the brain. So, and there are cells I cannot see actually. So I as much as possible. So I cannot tell you what those slices are, the small size, but these are all the gap here. They are all water, and you can see surrounding the here. You can see they are all water because there's a neuron. Around the neuron there are some astrocytic N feet they call, like the process go and they cover the neuron. That's the place they swell actually. These are the neurons, surrounding neuron. They are all the water accumulated in that cell. Yeah, next slide, please. Yeah. So can you expand the picture a little bit, whole screen or half screen? I'm seeing a stamp size, so I cannot see clearly. This is a stamp size picture in my computer. Okay, so I will continue. whatever I have, I will continue. I may or may not be able to give you too much information, but I can continue. So the previous one I showed you, there are swelling, right? It's acute condition. So what happened in chronic conditions? The chronic conditions, those cells not swell. So instead of what happened, they look like they are empty space in the middle. Can you see this one? They are all empty space around that area, and there are some small the brown spot. Can you see that? They call lipofuscin pigment. The lipofuscin pigments are they kill the cell. So they call us the oxidative marker. So they kill the cell. So basically, these cells die in the chronic conditions instead of they swell, and they don't die in the acute conditions. Simply they swell and they expand the brain and they die. But here in the chronic conditions, what happens? The cells die because they're getting some problem. They die. So there are two cells. Can you see that here? One big cells and small cells. Both are astrocytes. That's what I told you. These cells are always two. That's why they call us Alzheimer type 2 astrocytes. So here you can see, the chromatins are expanded. That clearly says that the nuclear membrane are damaged. So here, I'm sorry, I cannot see the slide carefully, but this one, is still you can see there are two cells in many places, but here there are only one. I cannot see the cell actually. So you may have to enlarge it in a better explanation. Maybe go to next slide. Maybe I may be able to see it there. Excuse me, can you go to the next slide so that, here I can show you even better here. So can you see that here there are all cells are two. Can you guys see that two cells everywhere or no? So someone say yes or no?
Yes.
So can you ask to expand the picture so that I can present a little better?
Okay, I'll ask Bala, then you'll continue, Jayakumar. You continue.
Yeah, okay. Here, in this slide, because I cannot show the cells how they look like, So here, in this slide is from, this is from rat brain, actually. This is a beautiful picture I took a few years ago. So what happened in the rat, we injected a liver toxin called thioacetamide. Here you can see there's a thioacetamide. If you inject that one, they kill the liver. So it creates exactly what happening in the human. So when you inject this liver toxin, you see, but very low dose for longer term. So you can see that two astrocytes together. But that is not occurring in the control. This is a normal brain. The cells are independent and there is no any two cells. Yeah, next slide, please. Yeah. Okay. This is one of the beautiful picture. This picture is one of the journal which I'm mentioning here. I'm not able to read. I think this is a Journal of Neurochemistry. This is the cover page in the journal. So one of the best pictures selected for the journal cover page. So here, see, this is a normal cells. This is one single astrocyte I took from the brain, which has liver here. This is a particular protein. There's a protein called thrombospondin 1. This protein. So you can see it dotted everywhere. It's sitting on the top of the cell. But when you go into the treatment, the animal which I treated, those proteins are lost here. You can compare from here to here. Those cells the proteins are lost. That's what the finding we had for the past 15 years. For some reason, the levels that particular protein is down. We don't know why it is down, but we are doing so many mechanic studies to see whether increasing that protein level can reverse the disease. So this is one of the best pictures we took. This is that astrocyte. So here the green is that particular protein. So next slide, please. the red here is again the same thing I told you. That's a GFAP, glial fibrillary acidic protein. When you measure the two of them, you can see a lot of yellow here, but there is no yellow here that indicating that that particular protein is lost. So please, next slide, please. Yeah. Okay. This is one of the beautiful picture I took. Actually, this was published in one of the major journal, and this is a great finding from my side. So this paper was accessed by several thousand people actually. So one of the best research so far. So this is what is this? This is a brain tumor. This is a brain. The two slides are from brain tumor. This slide is just like 1.5 centimeter away from the normal brain. You know, within the same area, like the tissue is together actually. This is 1.5 centimeter away. So you can see here, yeah, beautiful astrocyte. These astrocytes are bigger. Yeah, the screen is disappeared. Okay, I'm sorry. Okay, so okay, thank you so much. Yeah, it came again. So see, here you can see those astrocytes are small. This is a normal astrocyte in the brain, but when there is a tumor, they have the cells. You know, they are like buttons here. They are all attached to the dark blue color all neurons. So they are all connecting here. Look like a spider. You know, look like a spider. So what happened? These protein levels are increasing. The HEPATIC protein is increasing when you compare with the normal what the disease has. That means if you have tumor, those very different. They call us these cells call us reactive astrocyte. They respond to some insult. So the tumor is not normal cells. So those cells try to respond to any stimuli. Like if you hit something, if a dog or something, if you hit, they make noise. Those kind of expression the cells do when there is a tumor. So this is a beautiful picture I took, and it's published in one of the top journal. Here, this is in the liver problem. You know, the normal just simply liver problem. They are lost actually. This protein is lost, but here this protein is increased. That's a difference how these cells are behaving in each conditions. I mean, different diseases. Yeah, next slide, please. Okay. So this is okay. Then what happened? So we were examining why these cells, you know, what exactly happening in the cells. So why those cells become very different than normal cells. I mean the type 2 astrocyte, there are 2 astrocyte. Why they are 2 astrocyte? What exactly they do? So far there is no literature, no experiments, no evidence. No one shows what exactly the cell is, whether the cell contribute to the disease or not. We found one of the very important protein which is essential for the cell in one conditions. The other conditions that cells also what they do, they also give some trouble to the cell, like they create inflammation, they create a cell death process, all those things, and they kill the cells. Very interesting here. See, this is the animal which we treated with the liver toxin. The green here is the GMF in the normal brain, and this is the brain from the liver toxin treated. So what happened? The protein called glycogen maturation factor, the protein is increasing. You can see it increased many-fold. You know, that's what we found. So this is the red vitamin synthetase. That's the marker for astrocyte. This protein not changing, so we're using this one, that GFAP. So here you can see how much protein increase in the liver failure brain. So next slide, please. Okay, I'm sorry, I cannot see. So okay, the previous one. Okay, I'm sorry. Go to the previous one. Go to the previous one. I cannot see the slide. That's why. Previous one.
Excuse me, sir. Actually, who is sharing slide? I do not know. I am not able to see the slide, sir. Actually.
Okay, can you see now? Hello.
From who?
Hello. Hold the slide. Hold the previous slide, please.
Kindly pin your,
Okay, so the previous slide which I showed was a human brain. That's not rat brain. That was a human brain. Yeah, this is a human brain. Maybe you may be able to read here. This is from postmortem. This is from postmortem samples. This is not a rat. This is from postmortem sample. So here you can see the cells are so big here. But when you go to the next slide, that is in animal. I think I mentioned to you before, the human cells are so big than the rat. The same thing, we found in many occasions. Here, this is a normal. That's a normal human. I mean, the normal means you may be able to ask me what is normal. We take brain from trauma patients. They die within a few hours. So that we use as a control. So this is basically a person who died with the trauma. He died within 2 hours, and then we compared with the brain from HC for many years. Like, I think this person is 32 years. He has a liver cirrhosis. This is the brain. So you can see how much increase there that happened. The next slide, please go to next slide. The next slide is the rat brain which were treated with the liver toxin. So here, let's see, you can see still the levels are increased compared to control. The liver toxin, there is a lot of the protein. So we are still examining whether the protein really contributing or not. So in subsequent slide, I have some inhibitor which I used to suppress this protein to see whether that protein can improve the animal behavior. I will be showing that, but still, we are using a lot of like a knockout mouse and other things to see whether really this protein is the protein is bad for the animal. So that has to be confirmed. So next slide, please. Okay, so this is so interesting. So I showed in the one of the picture. I mean the HNE image. So the nuclear membrane is expanded, and then on the top of that there was a lipofuscin pigment. That what does that mean? Why they expanded? So it is possible what we thought. So the nuclear membrane may have some problem. So we quickly examined the lamin AC, because the nuclear membrane is the lamin AC protein, purely the lamin AC protein. So here you can see this is a normal brain. You mean this is normal brain. You can see that protein is located around the nucleus. But here, when you see in the liver toxin, those proteins are not. Here they are all separate protein. You can see like a dot. Some you can see inside the nucleus too. But here what happened? They are all aggregated. That's the problem. So once aggregated, what happened? They start to kill the cell itself. That's what happened when the lamin is aggregated. So it seems, so the aggregation of this lamin protein, so that's the cytoskeleton for the nucleus, right? So that may be the reason why those cells become like empty in the center, or something there. Something is not there that we are still examining. But this is a possibility that the cells become, the lamin become aggregated, and that may be the one giving some trouble that we are still examining. So we don't know what exactly this aggregated protein do. Yeah, next slide, please. These are also findings which some was published, some not published, but I'm bringing some most interesting aspects so that you guys can enjoy what's going on. So here, I think I mentioned before, in acute, nothing happened to the astrocyte. They simply swell and then they expand the brain and they die. But in the case of chronic, this protein going down for some reason. Here you can see the GFAP protein, they are down. This is simply the Western blot shows the two are from normal brain, and these are all basically cortical sections. So that someone may ask, what happened in other parts of the brain, like hippocampus or pons medulla or wherever it is striatum, wherever it is. But we analyze every area, but most important and exciting changes we observe only in the cortex, in particular in the gray matter. So we don't see that much problem in the white matter, but we don't know why. But the major changes they all occur in the gray matter. It is possible because it's a lot of astrocyte in the gray matter that is possible. Next slide, please. Okay. So, we did some behavioral study. This is hard to do. We did like a water mass to see how those animals behave when there is a liver problem. This is not a severe problem, as I said again. This is like chronic, mild and chronic. It stays for many years. These rats, we keep for longer, and we see they are all defective. This is two different measures we used the water mass. One they took, the path to reach. The other one is a spatial distance, how much they consumed. So very good. Yeah. So here you can see they show both measure. Yeah, both measurement they show changes. And when I used, actually what happened, I treated with because I mentioned to you before, there are a lot of oxidative stress markers are upregulated on the membrane. So we used N-acetylcysteine to see whether that can protect actually. So indeed we found in both the cases, both the different the parameters we measured, the NAC is improving. Actually, I told you there is no medicine or something, but in the chronic stage, if you go to hospital with the chronic, so they treat with NAC, but still they are not recovering actually. So as far as I know, until today, there is no treatment for both of the conditions. So they give rifaximin, it's like antibiotic, and they give all those lactulose and other things, but none of them work so far. So next slide, please. So you keep it here. Don't change it. The previous slide, which I said that the NAC work. So why it work here? Why is not working in the human? That's very, very interesting part I found. When I give NAC, it's not entering into the brain. This is the blood-brain barrier not allowing the NAC to go inside the brain. But in the case of when I give a derivative like NAC amide, they go inside the brain. But they work, functionally they work the same, and the NAC amide is working. But so far, people never check NAC amide for some reason. I have no clue. So we are, actually we patented it for treatment purpose for this particular disease. But this may be extended to other diseases like Parkinson's disease and other diseases also. But we patented it, and this has to go through many trials. Like they have to clearly check preclinical testing, and they have to go to phase 1, phase 2, and phase 3, and to confirm whether they are really safe for to the patient, and then they can probably try for the treatment. Next slide, please. Okay, it's hard to see, but okay. So here I was trying to show some of the inflammatory factors in the liver toxin treated, like a liver failure animal. So there are some changes. I have a list of protein here. I think they are all prostaglandins and other COX2, other markers. They are all changed in the liver toxin treated animal when you compare to control. So this picture is the quantitation for this one. Maybe you guys can share the picture later on and see, but not to use anything, but you can share and see what's going on. Next slide, please. I cannot see those, but I'm just assume that I'm talking correctly. So this one is okay. This one very important protein we found. I was mentioning you that there is one protein that levels are down called thrombospondin 1. So we went deep to check why that particular protein, the thrombospondin 1, is going down. So there are literature and there are so many studies in other conditions, not particularly this disease. In other diseases, they show that this protein, the c-MYC, this is one of the tumor marker. So when this is and transcription factor too, when this protein is more, it inhibit the thrombospondin 1. So then we checked what happened to this protein. This is a very interesting. This is a human. This is a postmortem sample. Again, this is not a TA treated animal. So when this is an, I guess this is a normal patient. This is a patient with the HG. So here you can see almost nothing in the normal patient. The normal again. This is not the trauma patient. This patient brain I took from the ischemia. So the person who died with ischemia quickly, I don't see any c-MYC, but when I took with the HE, I see a lot. But one thing I have to be honest with you guys here, ischemia. Again, the ischemia may not happen immediately. That might happen for a little bit time, and there are a lot of similarities between ischemia and the disease which I'm talking about, hepatic encephalopathy. But I don't know why this protein is not increased in ischemia. But yeah, again, these are all different diseases. But at least in this disease, I see increased c-MYC. That may be the reason why the TSP1, the thrombospondin 1, that particular protein located on the top, if you have like a cell here, and on the top, they are all called matrix protein. They call cell matrix protein. So whenever they need it, they go to the other cells and give communication. For example, from the astrocyte, they go to the neuron. In the neuron, they bind with the receptor, whatever is there, and then they give signal to the neuron to work better. That's the way it works. So here we see this the c-MYC protein. So that may be the reason the TSP1 is reduced in this cell. So next slide, please. These are all the These are all the astrocyte marker. Next slide, please. Oh, oops, I cannot see. Okay. So, okay, what happened in this cell? Again, these are also from postmortem samples, actually. So, how we have a lot of postmortem samples in our brain bank. We have a brain bank. Almost with the HCA disease, I have 15,000 brain. So, collected for more than 70 years. So, a lot of people whoever went before, they all collected. And one of my boss, he's a neuropathologist also. He also collected so many thousand brains. So we have so many brains, and we have access to all those things. So we see these cells, they express more TNF alpha. So it is possible that they are all inflammatory cells. So they produce a lot of cytokines, and those cytokines may have bothered all those things. That we did some study whether the cytokines can play some role, which I said maturation factor or other factor, and we found there is some information. So that I will present later. Next slide, please. It's a big slide, but I'm sorry. So this I have in my presentation. So this one, okay, here what happened. So I told before, the disease start with ammonia, because the gut derived ammonia, they are not metabolized in the liver. So they go to the brain and screw up the astrocyte. That's the way the disease starts, right? So here, okay, our lab has a model. We take a astrocyte from mice, rat, human, other higher animals, and we grow in our lab. If you go and see our publications, we have lot of paper which we use cultured astrocyte. I mean, not cultured, astrocyte cultures. We use a lot of astrocyte cultures, and when we write, very interesting, we see exactly what happened in the human brain. They start to swell. Actually, we measure, on time. So we see the cells start to swell bigger when you add ammonium chloride. So that's the way we test all those mechanisms, what happened, because we may not be able to see every